Neovascularization, the generating of new blood vessels, is a highly regulated process in both normal and physiological events. To better understand the neovascularization, it's important to reconstruct the process in natural cellular microenvironment in vitro.
We developed a microfluidic chip and automatic control, highly efficient circulation system to perform perfusion microtubes and simulated protein to recapitulate the initial event of neovascularization.
The microfluidic sprouting chip was consisted of 3 channels. An endothelial cell culture channel and a liquid channel were separated by a wide central hydrogel channel. The microfluidic control system was consisted of a micro syringe pump, an electromagnetic pinch valve, a bubble trap chip, a microfluidic sprouting chip, a microperistaltic pump, and a cultural medium reservoir.
With a microfluidic system, endothelial cells could be stimulated by high luminal shear stress, physiological level of transendothelial flow, and virus vascular endothelial growth factor distribution simultaneously.
Pipette 20 microliter of 125 microgram per milliliter fibronectin into 1 media injection port of cell culture channel. Cut a pipette tip to fit the port of cell culture channel with scissors. Insert the pipette tip into the other media injection port of cell culture channel. Then pipette out air from cell culture channel to fill it with fibronectin.